We'll be looking at the benefits and use and limitations of the COVID-19 testing methods. We'll be talking about several of the therapeutic applications for COVID-19, their treatments, and then finally, we're going to discuss some of the FDA emergency use authorization guidelines and how they apply to pharmacists during the COVID-19. But first, start out, what is coronavirus? Basically, the coronavirus is a large family of viruses that can cause illnesses in animals or humans. And in humans, several known coronaviruses can cause respiratory infections. And we've heard of the MERS, the Middle Eastern Respiratory Syndrome. We've heard of other viruses causing a range of colds to more severe diseases such as SARS, which stands for Severe Acute Respiratory Syndrome. And then, of course, the coronavirus disease, which is the COVID-19. It was first identified in China in 2019. And thus the name COVID-19 is caused, the COVID-19 disease is caused by the virus SARS-CoV-2. So it's the severe acute respiratory syndrome caused by the coronavirus 2. Early in the outbreak, many patients were reported to have a link to both the seafood and the live animal market, but later, there was no link to that market indicating a person-to-person spread of that disease. Also, the travel-related exportation of the cases have been reported. I tried to include the source documentation for the information at the bottom of each of the slides. So when you go through the information, the source documents were referenced in the lower bottom of the slide, and we'll be glad to provide you the original articles if you're interested. Just email us and we'll be glad to give it to you. Okay, question 1, which is the following is true? The disease is MERS from the virus SARS-CoV-2, or the disease is the COVID-19 from the virus MERS, or C, the disease is SARS-CoV-2 from the virus COVID-19, or the disease is the COVID-19 from the virus SARS-CoV-2. And the answer is D. So let's look at some of the actual testing. And we'll talk about not just the types of testing, but some of the applications, interpretation, site of collection, and the importance where you are in the disease state will dictate the positive or the negative response for that testing. We'll talk about some of the sensitivity, specificity, and what that means. And I wanted to bring that up and try to explain that because that's a very important gold standard that's been used by the FDA for many, many years for diagnostic tests. And we'll look at these in regards to the real-time PCR, or some people refer to it as NAT or nucleic acid amplification test. We'll look at some antigen and then also the antibody testing. So what do the diagnostic tests for COVID-19 actually detect? The diagnostic test for COVID actually detects either the virus or the immune response. And in the virus, it's actually detected by that nucleic acid amplification test, or the PCR, real-time PCR, which is used often in the sequencing and other genetic tests for looking at biomarkers or indicators. Also, the COVID-19, the virus is the viral antigen. And so you look at the structure there and you can see that nucleocapsid protein N, or the spike, and we'll talk about that. But those are some of the key components of the coronavirus that is referred to in the literature. The spike, or the protein, the envelope protein, which is noted here by E, and then the membrane glycoprotein. Or some of the tests are actually looking at the antibodies, which would be IgG or IgM or even IgA. And the thing to remember on the immune response, or looking at the test is looking for the antibodies as an immune response of the individual. Okay, so you're looking at IgG and IgM, and it was initially reported, and I tried to present data both for pros and cons of each of the issues, including the limitations of each, and then presenting that information to you. But the molecular test, the timing of the testing is really important. So you'll see many variables or many factors in this equation that are affecting the results of that test. So here's a good example, and of course this came from World Health Organization, where the individual will become infected. You have an incubation period, which may vary. And then the actual height, you'll see the symptoms occurring there in the middle part, where the molecular testing, which is actually detecting the virus. And in that stage, we pick up, most often, that's the real-time PCR is utilized to pick up the infected, or the molecular testing, the actual detecting the virus load in the individual. And so denoted by the blue line is that virus is detectable. And notice here, this slide is showing the early antibodies, IgM. The IgM is considered to be the antibody response to COVID, to the coronavirus, initially that's being produced, and then followed quickly by IgG. And it's believed to last much longer in the timeline. So you can see at which time you're testing an individual, whether it be asymptomatic or symptomatic, it's going to have an effect on that, on the result of that test. Okay, to diagnose an acute COVID-19 infection, that molecular test is used to detect that genetic material of the virus present in those samples coming from that nose, throat, or what we call the nasopharyngeal swabs. An antigen test might be used in the future to detect antigens, which are part of the virus. And currently available tests which need to be validated before use because they may miss people with active infections. And I know several individuals that have labs that are doing testing for COVID-19, and the antigen is not believed to be as reliable or as test as that molecular real-time PCR test. I think you will see some being developed, but just right now that molecular real-time PCR test tends to be the most frequently utilized test and relied upon. Okay, for serial surveillance of COVID infection, a serological blood test is used to detect the antibodies, as we just talked about, the IgG and IgM. So if you have the antibodies, that serological blood test is detecting IgG or IgM in response to those infections. And these antibodies last in the body for some time after the infection has passed. And those tests can inform you if you've been infected in the past or not. You know, it's interesting. I wanted to bring up another point here. There's a lot of data that says that the amount of time that each respective antibody lasts in an individual's is really not clearly understood until more studies are doing. And so it's not uncommon to have a false positive or a false negative when administering the antibody test, but it's frequently used for surveillance or the serial surveillance. And I'll talk later, a study we did with Stanford and published using the antibody test. Okay, I wanted to show this slide, and this is an important slide in that I talk about the condition or the disease. If you have the condition or have the disease, let's say you have COVID-19, you really have it. And the test, regardless if it's a rapid test, it's a real-time test, it's an antibody test, and the test reveals a positive result, then that's known as a true positive. If you really do not have the disease, but the test result indicates a positive, that would be a false positive. Similar logic would follow if you have that disease state or you have COVID and the test result shows negative, that would be a false negative. Likewise, if you do not have the disease and your test is negative, it's a true negative. And so ultimately, we are hoping to see positive, true positives are truly reported as positive, true negatives are reported as true negatives. Many compounders may have seen a similar situation in a clean room or a microbial investigation where they may have a false positive or a false negative. So it's really important to understand these parameters and this gold standard, because it's been utilized by the FDA and the clinical diagnostic world for years. And particularly, we use it to define the sensitivity and specificity. So when we talk about a sensitivity, that sensitivity is really looking at the true positive, the ability to detect true positives, and that's divided by true positives and false negatives. So remember that false negative, that person really has COVID, it just was reported negative. So you want to see how sensitive can you get in the assay. Specificity is the true negatives. So that's known by this slide, this quadrant, when the specificity of an assay or a test is if you're wanting to say, are you able to pick up negative? You don't want something that is not real that's giving you a signal. So of course you're wanting to measure you don't have that disease, and the result of that test, regardless of that assay test, is telling you that you don't have it. And you're doing that calculation. Now, a portion of this, it's really important to look at that positive predictive value and the negative predictive value. So those are very important to look at when you're looking at the ability to predict a population or prevalence of a disease state, whether it's symptomatic or asymptomatic. So that this group of parameters is an important aspect that the FDA and many individuals are used. Even in the analytical, we'll often look at a sensitivity and a specificity of an assay, as we just discussed, even in microbial. So these two terms are extremely important in the testing arena for COVID-19. The summary, I selected this one from the infectious disease to show you the summary of the findings of the test accuracy results for the prevalence or the pretest probability. I know this is a lot of numbers, but what I'd like to get from here is you can see that different samples taken from different sample sites, oral, nasal, the nasal pharyngeal. 2 studies were from the nasal and the saliva. A lot of work is being done in the saliva, and I'll talk about it later in the discussion as well. And then the mid-turbinate, which is in the middle of the nasal cavity, just a little bit up in the middle. And then I want to disclose the limitations. So the blue box to the right is really some of the things this study and this literature, when you look at it, it's really important. When you're evaluating, often we just hear the positive. But you want to hear the truth. You want to know what's accurate, what's specific, what's sensitive, and then know what are the limitations of the studies. To have true science, the limitations are just as important to look at the study design and the number of the statistics. So as you can see, a saliva sample, the sensitivity is 85 on these samples that were tested, and the specificity was 100. So you can see that the oral samples versus a nasal or mid-turbinate, which sample would you want to use? And then look at the consideration, the certain of evidence, the typical scene in symptomatic outpatients who've not reached a hospital facility. But it's important to look at all limitations when looking at a study. And so I wanted to just bring some general, there's no way we can get so granular in each of these areas today, but I wanted to just gloss over some of the main topics in this testing arena and therapeutics, and then the FDA EAUs. Okay, I thought this was an interesting slide because this came from the New England Journal of Medicine. And again, the source documents or the citations are at the bottom of the slide. But a false negative would mean the report is negative, but the patient really has a positive. So the clinician's perspective on challenges and implications regarding that. So what are the chances of the SARS-CoV-2 infection giving a negative test according to that pretest probability? And so this was a good slide just to demonstrate the greater the sensitivity And specificity, notice this first test has a sensitivity of 70, where the second one, the green line, has 90. Well, that again has a greater chance of infection and being accurate. So you can see the greater the viral load, the greater the chance of it. So multiple factors will be important in determining the test. The test is a measurement, and we often think of what is the cutoff, what is the result of a test. But it's not just the number, but what's the interpretation of the number? How do you apply it? And then what's the limit of detection, limit of quantitation? I mean, there are a lot of other variables without getting into it, but those two variables of sensitivity and selectivity and probability are extremely important, or predictive values are very important in looking at these, and these are considered gold standards. The FDA should ensure the manufacturers provide the details of the test, clinical sensitivity and specificity at the time. And the test without information will have less relevance to the patient care. And I thought this was an important also. Negative results, even on a highly sensitive test, cannot rule out infection. If the pre-test probability is high, so clinicians should not trust unexpected negative results. So it's important to understand the interpretation. What is the test? What does the test mean? Ask that question. If you as a patient, are taking a test, what is that sensitivity and specificity of that particular assay or test that you're taking? This slide I thought was also a good slide. We're looking at the estimated variation over time in the diagnostic test of the SARS-CoV infection relative to the symptom onset. So, I think it's nice to show a picture that shows 1,000 words. Basically, you can see the symptoms here. This is even before the symptoms start. The SARS-CoV virus is present. The actual symptoms begin, designated by this dotted line. And then you can see after the symptoms begin, you're very likely to have a PCR positive. So it is possible or plausible that if you have a test by real-time PCR, it comes back negative, you could either be way early or way out here, when it would not be positive. You can see it could be negative. So if you're asymptomatic or symptomatic, you're going to see the difference. The timing is very important when a test is being done. And I thought this was really good. Even the color, again, this is an excellent one. This was one of my favorite slides of the whole presentation from JAMA. The nasopharyngeal is this blue slide, and you can see that's considered probably one of the most accurate by PCR real time. Also, some people will call it NAT in the literature, standing for nucleic acid amplification test. But notice the virus isolation from the respiratory tract, very quickly up and down, even within the first week. PCR likely. So that based upon the sample collection site, it's going to dictate how long it would be able to be detected by PCR. And then notice an antibody, if you test too early in the cycle of the disease state, you're not going to pick up the antibodies, but not until usually a week or so later. And I wanted to show something here. Early on, previous slides, I showed notice it was believed that the IgM was formed well before the IgG. Now, in the more recent data, you're actually seeing in the literature fairly close. Even I read an article just yesterday where just saying the day, hours. But I don't know the truth of that. But I'm presenting the data as it's presented in the literature. And so this is the literature saying that the antibody for IgM does form quicker than IgG. But I think one thing you can say for sure with high degree of confidence, that the IgM drops off quickly, whereas the IgG persists. Okay, looking at the real-time PCR testing, there are two frequently used type tests: the nucleic acid amplification test, as we just stated, and antigen-based tests. The real-time PCR test was first confirmed and developed by Corman in 2020. So Also, I wanted to give you the citations if you're interested in the literature, going back when was it first started and reported. And then remember that the SARS-CoV-2 real-time PCR detects the viral load. So you're actually measuring the RNA. And a positive result is highly specific for that presence of that virus. So it's a very accurate for that particular virus. And the load, the sensitivity of these tests is not uniform and is affected not only by the assay itself, but also, as we were talking about earlier, the limits of detection, the viral inoculum, and also the timing of the testing, and even the sample collection side. So I think sometimes we think, okay, testing something, even testing a drug in a preparation, a product or a preparation, there are many factors that could affect the result. But hopefully you can understand. I thought Rick Rhodes did an excellent job on describing when he was talking about stability studies earlier and the various factors that enter into it. Okay, here's another good slide. This is actually from Lancet Infectious Disease, which is it's a pretty credible journal as well. I like this particular slide. This illustrates the temporal profile of the viral load of the virus here. And it's talking about saliva. And you see a lot of testing labs now will use saliva for the matrix or the bio sample to do the testing. And this was a cohort study from 2 hospitals in Hong Kong. And then these are the results illustrating that the viral load was highest during the first week after symptoms onsets and subsequently declined with time. And you can see that based upon this regression line. And again, this mimics and verifies the data that you see in the other literature that I presented earlier in the presentation. Okay, not only is it presented over time of symptoms, but also the viral loads with different tissue samples. And I thought this was a good one in clinical infectious disease. This one shows the first group, maybe you can't see it on this slide, but this is in the order of presentation. This is the nasal swabs, which is the rust. The blue-green is the throat swabs, and then the sputum. And then, of course, one would expect blood and urine to be much, much lower here from the viral load of the different tissue samples. So this was taken from about 323 samples in Beijing, and that was analyzed for 2 target genes listed here. But you can see they looked at margin of errors and also the probability. Remember from our statistics, the lower the number of the p-value, the greater the significance that one would expect. And so you can see that data that presented here. And again, over here, the right side of the slide, the analysis of the viral load in different clinical stages. So not only is the sample, so this slide demonstrates the sample that's selected that you're testing will affect the result of the test, but also which stage of the disease state are you in? Are you in the early stages, which you can see is best detected in the nasal swab, the recovery stage, which you can see that it's the throat swabs are decreased. But if you took at your measurement of air lines and you look at those, you can see the decrease. And then the clinical cure, which that data, you're in the late stage of the disease and you're seeing at the low level of the viral load present. Okay, rapid antigen testing. The interest in the rapid molecular antigen testing for SARS has been high since we started. And that makes sense. I mean, this is a major serious issue. And the quicker you can test and understand it, the better that we can to deal with it. So 2 major categories of these rapid tests are the rapid molecular tests and the antigen tests. These rapid antigen tests are most often antibody-based to capture that SARS-CoV-2 antigen. And typically it's the N protein. I wanted to bring that out because that's how it was designed or how the test was designed. And we illustrated what part of the protein on one of the earlier slides. And the other is the rapid molecular test detecting viral RNA. The sample type, as we've just learned, mid-turbinate or the nasal swab is very important rather than nasal pharyngeal swab. This was a very important, I thought this was a neat a bullet from the CDC. Important difference in rapid testing in particular is that some studies have shown that persons who have symptoms for more than 5 days may no longer have the antigen at the level of detection. So I wanted to say, particularly among asymptomatic patients, And I think this is what's presenting a problem today for us in the diagnosis and what stages of it is, and even in the infection period. Because I suspect a lot of people are walking around asymptomatic and they've never been tested. They didn't even have a clue of it. But based upon that study, we see, I'll talk about a study that we actually did with, on asymptomatic patients. But this is an issue here, actually. So the FDA has issued individual emergency use authorizations on several antigen and rapid molecular tests. Actually, just today, the FDA authorized the first test that detect the neutralizing antibodies from a recent or prior SARS-CoV infection. I think that was important. The FDA issued that emergency use authorization, which specifically tests this type of an antibody. And it's really detecting the neutralizing antibodies. And so it's not the standard antibody test, but the neutralizing antibodies test. So as we talked, so that was a real time, the antigen, and now we're talking about the antibody test. The point I wanted to bring up is that, remember, these are serological tests. They measure the immune response. So you're looking at IgG and IgM versus the viral RNA load. Again, there's several studies that are frequently cited in the literature, and I wanted to give the author and the location of that. And not all patients with SARS-CoV infection develop an antibody response. So that was in line with what we were talking about. You might take a test, you may have it, and you may not have that antibody response. So the negative serological result does not exclude past infection. So that would be another limitation, but one would need to consider. Lastly, and this is a very important issue, the sensitivity and specificity of tests are impacted by the community prevalence. So the more frequent an individual would have, that calculation would go into it when they're conducting those tests and reporting that often. This was a study that we actually did. We teamed up with Stanford and a group of scientists, basically in Salt Lake, and then Stanford. And this was utilizing those laminar flow, looks like a pregnancy test, that had IgG, IgM, or negative. And so this was used in several sites across the western part of California. And this was one that we studied. We actually published in the Cold Springs Harbor Laboratory in April. And we had a lot of criticism for the calculations of the statistics, which I think really should be considered in the publication. But this was actually, the primary author was from Stanford. There was a lot of people, MD, PhD, and public health experts involved in this particular study that we did with Stanford. But this was one of the original studies. And the important was, you're wanting to know what is the prevalence of the disease, COVID-19, in a particular area. And so often this, as we mentioned earlier in the discussion, the seroprevalence may be present. It may be a good tool to look at that. But unfortunately, you need to remember that population prevalence in the calculation statistics. Okay, the SARS-CoV. I wanted to talk about on this particular slide, look how easy this point of care, this rapid, it's usually handheld, minimal training is needed. We didn't put on here, but the absence or presence of their qualitative is more qualitative, excuse me, not quantitative. Versus a neutralization assay, which the lab requires at least a BSL-3. That's pretty critical. That's Biosafety Lab 3 levels if you're using a live SARS-CoV extensive training needed. So you can see there's a big variation of the test that's being used, the time that it takes to get the results, and what does it tell you, what does it not tell you? Okay, let's look at the second question. Of the following tests, which would you consider the most beneficial based upon the scientific data or sensitivity and specificity, and probably should consider the positive predictive value and the negative predictive value? Would you consider A, serological blood test to detect the antibody IgM, IgG? Or do you think a rapid antigen test would be more beneficial? Or do you feel that the viral RNA detected by real-time PCR, or none of these tests are beneficial. What do you think the answer would be on that one? Right, it's C, the viral RNA detected by real-time PCR. Okay, let's look at some of the therapeutic applications for COVID-19. Okay, we'll look at the convalescent plasma, hydroxychloroquine, monoclonal antibody, remdesivir, and then some of the other treatments. Okay, so the concept of basic convalescent plasma to treat COVID-19 is you're taking the blood from donors who've actually recovered from the COVID-19, and so that you have the donor apheresis. Of course, you're matching the ABO blood type to the individual receiving it, and then taking the plasma, which will be the convalescent plasma, and then administering it to that patient. So the idea is you're taking the neutralizing antibodies from the individual that has recovered from COVID-19 and infusing them into a passive administration into a patient, hopefully to give them that would find beneficial. And of course, I'm crediting the author on this diagram. But the SARS-CoV neutralizing enzymes here, neutralizing antibodies. Several studies. The benefit has not been consistently studied in a randomized manner. I wanted to bring that, and this just states what I've just indicated, that randomized antibody from a person is given from one patient to another who has not yet developed their own antibody response. Again, multiple studies are saying variable results. I wanted to bring this last comment up because it's really interesting. On September the 23rd of this year, the FDA issued an update on convalescent plasma therapy. But if you really read it and look at the basis of it, it says the update included analysis that supported the concept. They didn't come out strong. I mean, they said, and they even in their terminology said may be effective. And that's based upon several scientific information. So they're not coming out strong, I don't think, in supporting that, but you can see it may be effective. And I think this is further data that supports this. This was a study that was published in JAMA in the New England in 2020, and a multi-center open label randomized controlled study. And you can see there was no significant difference in improvement, 51 versus 43, or the mortality rate. And you can see that the control listed here and the convalescent plasma. Here's another study, the effect of the convalescent plasma. In this study, which I thought was a pretty decent study, but the conclusion, as in the meeting, it did not significantly improve the time to clinical improvement within 30 days. Here, the NIH lists the summary recommendations. Plasma from donors who have recovered from 19 may contain antibodies to severe acute respiratory syndrome that may suppress the virus and modify the inflammatory response. What I wanted to bring out here, again, notice there's insufficient data for the COVID-19 treatment guideline panel to recommend either for or against using the following blood-derived products for the treatment of COVID-19. So it's important to look at both the pro and the cons and the data and the data that can support that conclusion. Okay, let's look at hydroxychloroquine. Chloroquine and hydroxychloroquine both have that in vitro activity against the SARS-CoV-2. It's unclear if one is more potent than the other, but there are conflicting reports depending on the study that you read. The availability and the side effect and profile of hydroxychloroquine is favorable to the chloroquine. And these are some of the sources that we took that data from. Again, the conclusion, you can see an observational study of hydroxychloroquine in hospitalized patients with COVID-19. They were not associated with either a greatly lowered or an increased risk of the composite endpoint of intubation of death. And this was the randomized controlled trials of hydroxychloroquine in patients with COVID are needed. So they're stating that was back in May when this was cited. I wanted to bring this one up because later on I present some information with the hydroxychloroquine and remdesivir with the azithromycin. But in hospital mortality in patients with COVID-19 treated with hydroxychloroquine or azithromycin, the authors concluded, and this was taken from the JAMA just back in May, was not significantly associated with differences in hospital mortality. The early treatment of the COVID-19 patients with hydroxychloroquine and azithromycin. This is an interesting because I'm going to show two citations that communicate at both sides. And I think it's important to present all the data that you see in the literature. Administration of the hydroxychloroquine and azithromycin combination before the COVID-19 complication occurs is safe and associated with a very low fatality rate in patients. So they're saying administration before the complications occur is safe and associated with a low fatality rate. So it may be at which point you're administering that. And I thought this was an interesting, that impact of hydroxychloroquine with and without azithromycin on those SARS-CoV-2 load. And you notice this result. The blue is the hydroxychloroquine, the brown is the hydroxychloroquine with the azithromycin. And then the green is the comparison. But I wanted to bring out, this slide is the negative. So it's the inverse, it's the reduction or the disappearance of the COVID-19. And so you can see, despite its small sample size, our survey shows that hydroxychloroquine treatment is significantly associated with viral load reduction or disappearance in COVID, and its effect is reinforced by azithromycin. So the conclusions of this journal is showing when combined, the hydroxychloroquine, when combined with azithromycin, and you can see that in the reduction, the increased reduction of the viral load in this reference. And then the lack of benefit with hydroxychloroquine plus azithromycin in 11 patients with severe COVID-19 infections. The authors concluded, in summary, despite a report antiviral activity of chloroquine against COVID-19, we found no evidence of a strong antiviral activity or clinical benefits of the combination of hydroxychloroquine and azithromycin for the treatment of our patients. So I wanted to bring this out because the FDA actually revokes the emergency use authorization for chloroquine and hydroxychloroquine in June of 2020. So this citation, you're looking in the literature and you're looking at it. So that's why you might see different articles cite different conclusions, but it's so important to have a double-blind, controlled, randomized study with as many data points as possible. Okay, moving to the antibodies. This was just a picture illustrating from NIH talking about the antibodies binding to the surface of the virus, blocking entry into that human cell. So we can see the antibodies. And actually, I'm going to Regen just came out with the notification. I'll read that when I get down to the antibody section. But just today with it. But monoclonal antibodies are often created by looking at that pathogen-specific B cells to the patients. Let's cut to the chase. I mean, what I wanted to say is monoclonal antibodies have been used for other treatments. I mean, we've used it for HIV, influenza, RSV, the MERS. We talked about that Middle Eastern respiratory syndrome, coronavirus, Ebola, and also Zika virus, as stated in the study by Walker. The point I wanted to bring out is Eli Lilly and Regeneron Pharmaceuticals. In October 2020, both companies applied for emergency use authorization from the FDA based upon internal results from the trials in progress. Eli Lilly's monoclonal antibody regimen consists of 2 antibodies directed against the SARS-CoV-2. Remember, we talked about the spike protein and the receptor binding domain early on, and the Regeneron product, the Regen-CoV-2, consists of 2 binding to different regions of the SARS spike protein receptor. Now, just today, it just came out this morning about 9 o'clock, Regeneron predict 300,000 COVID-19 cocktail doses ready by January and substantially more in 2021. And this was released by Fierce Pharma Manufacturing. It was released this morning on November the 6th at 9:18. And it says that now that an antibody cocktail has shown promise, and as the raging pandemic hits new heights, The U.S., the company is amping up its efforts to manufacture hundreds of thousands of doses. And we get this question frequently, when do you think it's going to come out? Or what do you think, Tom? And, they'll ask, what do you feel the status and how quick are we going to get a vaccine? And it says that as Regeneron awaits word from the FDA on its therapeutic, the company is producing hundreds of thousands of doses by January under supply deal with the U.S. government. The company expects to have 2.4 gram doses ready for about 80,000 patients by the end of November and 200,000 doses ready by the first week of January, according to the chief scientific officer. And then by the end of January, Regeneron is anticipating to have around 300,000 doses ready for its use. And so I think that's interesting as we talk about monoclonal antibodies in this prospect, in this context. This is a picture I took directly off I was watching the news. Caroline was watching the evening news, and it came up with the president. We all know was in the hospital, and it stated he was given a polyclonal antibody cocktail as well as the zinc and vitamin D, and then what else he was given. So this was on an ABC News. I don't remember that it was the night that was on. I just took a picture of the TV, and then I thought it'd be nice for this presentation. But, in October, of course, we know he received this Regeneron pharmaceutical experimental monoclonal antibody, Regen-CoV-2, as part of his treatment for that COVID. And then both the Regeneron, so that's stating just what I just got through reading from the information that came out from Fierce Pharma today. What's the deal? Okay, yeah, this was a good article by Chen. This is SARS-CoV neutralizing antibody in outpatients. So here's the conclusions from the antibody, these antibody. One of the 3 doses of neutralizing antibody appeared to accelerate the natural decline in viral load over time. And so here that's funded by Eli Lilly. That just came out last week. I wanted to present sort of the latest data because this is changing rapidly. Okay, remdesivir. Here, this study came out by ASHP on Friday, October the 16th, and saying it's not been yet peer-reviewed or published. The only antiviral drug authorized for treatment fails to prevent deaths among patients, according to it. Gilead disputed the conclusions, noting a variety of drugs and drug combinations. And I think I wanted to bring that attention to drug combinations. Remember, we talked about the azithromycin, and then further, a later slide, you're going to see it with dexamethasone and the importance of them working together. Has been evaluated under a wider range of circumstances. Okay, generally, there are 3 bullets I wanted to bring out from remdesivir. It's the first drug found to have beneficial effect on the hospital patients with COVID-19. Early in the pandemic, the remdesivir was found to have antiviral effect against the SARS-CoV-2 in both in vitro and in vivo studies with the rhesus macaques. And that was well cited in multiple places. And then in May of 2020, the FDA approved that emergency use authorization for it in adults and children hospitalized with severe COVID-19. In summary, that remdesivir does not provide an overall mortality benefit to the general group of the patients, but it does reduce the time to clinical improvement when given early in the course of the illness and or patients with less severe disease. We can't have a presentation without a molecular structure or a compound, so we put this up here from PubChem, but that's the structure, chemical structure for remdesivir. And bottom line is, again, October the 22nd, the FDA approved it for use in adults and pediatric patients 12 years of age and older and weighing at least 40 kilograms for the treatment of COVID-19 requiring hospitalization. And then I'm saying very similar to this. And so basically the FDA, EAU, was adjusted to continue that emergency use for hospitalization for hospitalized pediatric patients. How am I doing on time? Okay. This slide I thought was interesting because it shows the escalating phases of the virus disease as it progresses through time. And so this disease progression with associated signs and symptoms, and then the potential phase specific therapies. And so we took this from this particular journal. You note the symptoms and then the clinical signs and then the potential therapies. All throughout here, you're seeing the treatments that I've listed here today, the remdesivir, the chloroquine, hydroxychloroquine, and the convalescent plasma. But notice here in this phase 2, pulmonary phase, the careful use of corticosteroids, other immunoglobulins, and inhibitors. So you're seeing the use in combination with these other treatments. And I wanted to bring that out in this particular slide. Okay, the NIH guidelines, therapeutic management. I think this was a pretty decent little graph in that it talked about the disease severity, again, showing at which stage are you in the disease state and which treatment or therapy would you use. Initially, you're not recommending the use of dexamethasone, and then you see hospitalized or the oxygen. And this is according to the NIH therapeutic management guidelines. You see the remdesivir, and then later on, you're seeing the additional of the dexamethasone being added to that. And then hospitalization requiring invasive mechanical ventilation, and of course, dexamethasone being here. So this is just indicating some additional adjuvant study therapy. So question 3, based upon the scientific literature thus far, which would be the best choice for therapy for the COVID-19? Convalescent plasma, hydroxychloroquine, remdesivir, or none of the above? C, remdesivir. Yeah. Okay, let's look at vaccines. We can't all ignore vaccine. And there's an extensive pipeline for that, from the exploratory to each of the individual phases and then the actual aspect of it. The 3 phases is the phase 1 of a clinical trial, of course, is the safety. Phase 2 is the safety and dosing, and then it's starting to phase 3, looking at the safety and efficacy, and then all this data is compiled and given to the FDA for the review, whether they'll determine it is approved or not. But I wanted you to bring also the increasing number of participants as you conduct the study. So this phase is the traditional clinical trial phase that's incorporated, and you've probably seen similar slides before. But the increasing number, of course, we know that the more participants, the greater the data and the confidence you have in that data. Okay, vaccine development. This is what's amazing. Notice how many years from preclinical to the manufacturing process to the optimization of the assay. Then you go into so this is even preclinical before it's even started. Then you have the safety of the vaccine, then the safety and the immunogenicity, and then the safety, efficacy, and regulatory approval. So you can see it takes many years. So it's amazing that we're able to do with what we're doing just the short time from the initiation of the disease to the end. But I think it shows a good collaboration between the government entities, academic, between the private pharmaceutical companies, and all the resources that's been thrown to it to be able to get a vaccination by, I believe that you'll see it next year. Again, New York Times, this is just that came out recently and just last week. We took this from the New York Times. It was saying 36 that was in phase 1, 14 in that phase 2, down to right now there's none approved, but there's 16 that are approved for earlier limited use. So it looks very favorable that some will come through and that will be successful. And of course, we know there are multiple vaccines in being studied. Okay, this was just a slide showing all the different companies that are involved in the vaccine that are working with it. We know AstraZeneca, Biotech, Johnson, Moderna, it's been in the news a lot, Pfizer, Sanofi. So there's multiple ones. And then this is a good citation. They're all over. You can see the tracking. You can look at them. You can see the institution that's doing the study. So there's a lot of data out there in the vaccine trackers to see the state that they're in. This was a good slide. I like this particular publication. This was at Mayo Clinic, Poland, and this was talking about the number of the vaccine candidates. But what I liked about it was that it give you the various types of vaccines. It said more than 120 vaccines candidates are in various preclinical and phase 1 to 2 and 3 trials that included inactivated, live attenuated, viral vectored, replicating, and non-replicating protein and peptide-based and nucleic acid approaches. So there, the endpoint there, we could have vaccines that have different approaches, and I just wanted to bring that out. This was a good paper. It's one of my favorite papers regarding vaccine, and it came out from Mayo Clinic. Any of you want these data? I have PDFs of these. Okay, let's look at the FDA emergency use authorization guidelines and how they apply to the pharmacist. Again, it stands for emergency use authorization. During a public health crisis such as COVID-19, there may be an urgent need for this. This allows the FDA to have more timely access to drugs or the public, diagnostic tests, and other critical medical device or products. You know, when there's no adequate approved or available options and they cannot wait for all evidence needed for a full FDA approval, as we just saw in the phases, both from a vaccine and the clinical trials, it takes years. We don't have, when you're balancing the risk and the benefit to public health, it's important. Evaluate quickly using the available evidence. And this is, I thought this is very important. The FDA balances known or potential risk of the unproven products with known or potential benefits of the public during that emergency. Okay, these are some emergency use authorization. Here you saw the plasma back in August, remdesivir, certain patients, and then the compounding. Earlier, someone, I don't know who listed these, but these were listed on some compounding guidance documents that have been listed by the FDA, talking about hand sanitizers and other issues, temporary policy for compounding of certain drugs for hospitalized patients. So these are a source for you for that perspective. Question 4, the FDA, EUA stands for Food and Drug Administration, Emergency Use Association, true or false? Of course, it's false. Okay, we want to thank you for this time. And at this time, the claim code is umbrella. Umbrella is the claim code. Okay, I'll stop sharing my screen and turn it back over to the moderator. Thanks again. If there's any questions.
We have a question from Linda McElhaney. What do you think of the spit test that the NFL is using? Is it a type of PCR test too?
Well, I have to disclose I'm on a board with Smyrtle, so I may have opinion on that matter. Smyrtle's Sports Medicine Research Testing Lab, and they did the Major League Baseball, and then also been involved in the NFL piece. But we say the spit, but really the correct name is saliva. And yes, you see that, and I actually stated several of the reports, and I selected those articles just for that purpose, and you see the saliva. So yes, it is a good test. It is a good site, and you're seeing more and more tests being developed with that. And that's not surprising because, you know, a lot of the other cancer and other diagnostic tests, really it's a genetic, and so long as you can get that nucleic or nuclear material from the mouth and be able to extract it and analyze it, you can see it. So it is a good source, Linda, and that spit or saliva is a good test, and it's being utilized frequently today in many labs across the country.
We have a question from Gigi Davidson. Since natural COVID antibodies last only 3 to 4 months, what does this say about the duration of protection from a vaccine? Will we have to get boosted 4 times a year?
Oh, excellent question, Gigi. Of course, I have to disclose I have an inherent bias toward Gigi. She's incredibly sharp and bright. She's going to be speaking tomorrow. I'll do a plug-in for her. But Gigi, I don't think we really know. We say that they're only 3 to 4 months, but I'm not sure that they are. I know that actually, I believe Oklahoma Medical Research Foundation is doing a research project looking at measuring those antibodies and how long. I know people say that, but I think a lot of this is based upon your own immune response and how healthy you are going into the disease, how healthy you are through the disease and the treatment process. So the antibodies, you're correct that you hear there's a limited, but I don't think we know for sure how long we produce the antibodies, even how quick. And as the data that I brought it out early in the discussion, where initially we said IgM is going to start, then a week or two later, IgG. Well, we're seeing IgM and IgG probably is forming closer in the humoral response. But I'm not sure that we really know that it is 3, 4, 5 months exactly right now.